Good afternoon, students. In yesterday's class, I explained you about the two core techniques of biotechnology. Then I explained you about the conceptual development of principles of rDNA technology. Then I explained you about actually, I started the introduction of aligned DNA into the host organisms. Just briefly, I explained you about that. So now I will explain you in detail about that one. In the previous class, even I told you that who is known as the father of genetic engineering. The father of genetic engineering is Paul Berg. So Paul Berg is known as the father of genetic engineering because the scientist Paul Berg he isolated the gene from the SV virus 40, and that one he introduced into the bacterium through lambda phage. So for that work, even he got Nobel Prize also, and is known as father of genetic engineering. After that, actually I was explaining about the when aligned DNA, aligned DNA, aligned DNA, or it is called as the foreign DNA, foreign DNA, aligned DNA or the foreign DNA. See, this is the aligned DNA, or we can tell the foreign DNA. So this foreign DNA when this foreign DNA is introduced into the host cell, it introduced into the host. So it will not replicate. It is not going to replicate. Is it clear? Why this foreign DNA, when you are going to insert into the host, introduce into the host cell, why it not going to replicate? Because here this DNA they are introduced into the cytoplasm of the cell. Is it clear? When this DNA, when the foreign DNA, this is the nucleus, okay? This is the cytoplasm, this is the chromosome. You know very well, in the eukaryotic cell, we are going to observe the nucleus. So inside that nuclear membrane, we are going to observe the chromosome, which is made up of what? Nucleic acid, that is the DNA. That is the DNA. Chemically, you know very well. Chromosome, it is chemically made up of the DNA as well as the protein. Is it clear? So when the foreign DNA is integrated into, when this foreign DNA is integrated into the genome of the chromosome, when this foreign DNA integrated with the chromosome, it is going to what? Replicate, and it is going to what? Multiply. Because chromosome has specific DNA sequence. This chromosome has specific DNA sequence, or we can tell that nucleotide sequence. Is it clear? So this chromosome has a specific DNA sequence or nucleotide sequence, and that specific DNA sequence is called as ori, or is called as the origin of replication. It is called as the origin of replication. Is it clear? So this chromosome has a specific DNA sequence, and that is called as the origin of replication. Means this, through this, what will happen then? Replication is going to begin. Is it clear? When the aligned DNA or foreign DNA is integrated with this region or bind with this one, what will happen? It going to replicate, and it is, or we can tell that it's going to multiply. If suppose this DNA is introduced here, so it is not going to what? The replicate, but this DNA, when it is integrated with the chromosome, what will be happen? It is going to what? The replicate. It is going to the multiply. Because chromosome has a specific DNA sequence called as the origin of replication, and in that region, when they are going to bind this one, what will be happen then? Replication occurs. Is it clear? It produces the multiple copies of the DNA. So this is the brief introduction. Okay. After this, we'll go to the next one. Is the moving to the next one. Moving to the next one is the construction of first artificial recombinant DNA. Construction of first artificial recombinant DNA, or short form what we are going to call the rDNA. Recombinant DNA. Recombinant DNA. The first recombinant DNA was constructed by the scientist Stanley Cohen and Herbert Boyer. Herbert Boyer in the year 1972. Is it clear? First artificial recombinant DNA was constructed by two scientists. One is Stanley Cohen and second one is the Herbert Boyer. It is a very important who first constructed artificial recombinant DNA. It was first constructed by the scientists Stanley Cohen and Herbert Boyer in the year 1972. These two scientists, they belong to the United Kingdom. Is it clear? So Stanley Cohen and Herbert Boyer, they first constructed the recombinant DNA. Okay. Scientists Stanley Cohen and the Herbert Boyer. This is a very important concept. Just listen here. A bacteria that is called as the Salmonella typhimurium. Salmonella typhimurium. It is one of the bacteria. Salmonella typhimurium. It is one of the bacteria. This is the genomic DNA. And beside this genomic DNA, in prokaryotic cells such as in bacteria, extra chromosomal extra chromosomal self-replicating. Yesterday I was explaining you about the plasmid. Plasmid. See here the bacterial cell. This is the genomic DNA. Beside this genomic DNA, extra chromosomal, self-replicating, double-stranded DNA molecule present in the cytoplasm of prokaryotic cell. Yesterday already I told you what is a plasmid. Plasmid, it is a extra chromosomal self-replicating double-stranded circular DNA molecule found in the cytoplasm of the prokaryotic cell. So bacteria is also the prokaryotic cell. So here beside this genomic DNA, we are going to observe the extra chromosomal or extra genetic material that is called as the plasmid. Is it clear? So scientists Stanley Cohen and Herbert Boyer, so they are which bacteria they are taken, that is the Salmonella typhimurium. You know very well it causes typhoid. Is it clear? So this plasmid, okay, this is genomic DNA and this is the plasmid. Okay, so this plasmid have the antibiotic resistant gene. Antibiotic resistant gene. So this plasmid, it contains what? Antibiotic resistant gene. So this antibiotic resistant gene means it is the what? The DNA. So this antibiotic resistant gene, they are going to what? Cut. This antibiotic resistant gene is going to what? Cut with the help of enzyme. Enzyme restriction enzyme, or we can tell restriction endonuclease. Short form we are going to call as the REN. After this, I am going to explain you about this restriction endonuclease. So, restriction endonuclease, it is called as the molecular seizure. It is called as the molecular seizure. Is it clear? Because it cut the DNA at specific region. So, this restriction endonuclease, it going to what? Cut this antibiotic resistant gene. Okay, so this antibiotic resistant gene, this is the one, antibiotic resistant gene are going to this is the one, the antibiotic resistant gene. Okay, and this is the vector. This is the one, vector. Vector is also the plasmid is called as the vector. Is it there? Vector is required for transferring the DNA from one cell to the what? The another cell. Without a vector, it means that vehicle for transferring the DNA from one to the another, vector is required. Is it clear? Same thing, when the person will move from one town to the other town, how he will go? He will go in bus or he will go in vehicle. So vehicle is required. Is it clear? Same thing when the foreign DNA, this antibiotic resistant gene is transferred from this bacteria to the another. So what we require? Vector is required. So this vector is nothing but what? Plasmid. Vector is nothing but what? Plasmid. So same enzyme is going to use to cut that region. Here also they are going to cut with same enzyme, that is the restriction endonuclease, and in this cut region, so this DNA is going to what? This DNA is going to
insert. And this DNA is called as what? This DNA is called as the recombinant. It is called as the what? Recombinant DNA. It is called as the what? Recombinant DNA. This is the plasmid and this is the antibiotic resistant from which one? Salmonella typhimurium. From this they are going to insert into the vector and now it is called as the what? Recombinant DNA or it is also called as the mosaic DNA. It is called as the mosaic DNA or it is also called as the chimeric. It is called as the chimeric DNA or even we are going to call it is also called as the hybrid DNA also. It is also called as the hybrid DNA. Is it clear? So, this DNA is called as recombinant DNA or it is also called as the chimeric DNA or it is also called as the mosaic DNA or it is also called as the hybrid DNA. Okay, one more thing. Okay, I forgot to tell you. So, DNA is cut with the help of the enzyme restriction endonuclease. Okay, after cutting the DNA, so that part of DNA, how they are going to join? So the DNA is joined with the help of the enzyme ligase. With the help of the enzyme ligase. Is it clear? Restriction endonuclease, it cut the DNA but ligase enzyme, it joined the two sticky ends of the DNA. So because of that, ligase, it is known as the molecular. It is known as the molecular structure. Is it clear? Restriction endonuclease is called as the molecular seizure or it is also called as the cleavage enzyme. But ligase is known as the molecular structure or it is also called as the molecular glue. Due to the discovery of this out there, it is not possible to construct the recombinant DNA. Is it clear? So, due to the discovery of the restriction endonuclease as well as the ligase enzyme, so there is the construction of the recombinant DNA. Now, this recombinant DNA is transferred into the vein. So this recombinant DNA, which containing the antibiotic resistant gene, so that they are going to introduce into the vein. So this recombinant DNA is introduced into the vein, into the E. coli. Introduced into the vein, E. coli bacteria is called as what? Escherichia coli. Escherichia coli. So this recombinant DNA, where they are going to insert, that they are going to introduce into the vein, into the E. coli bacteria. Remember it here that antibiotic resistant gene is isolated from the what? Native plasmid of Salmonella typhimurium, and that antibiotic resistant gene is introduced into the vector. When they are introduced into the vector with the help of the enzyme ligase, that DNA is called as what? Recombinant DNA, and that recombinant DNA is introduced into the introduced into the E. coli bacteria. Introduced into the E. coli bacteria. here, this recombinant DNA, it is using this new host cell and it is going to what? Multiply. Because E. coli bacteria, it is a close relative of what? Salmonella typhimurium. Is it clear? E. coli bacteria is a close relative of what? Salmonella typhimurium. So, this recombinant DNA, it is going to what? The multiply. Understanding here? So, the replicate to multiply antibiotic resistant gene, gene in E. coli. The replicate to multiply antibiotic resistant gene in E. coli. Is it clear? So when it is transferred into the E. coli bacteria, so this recombinant DNA, it is when actually this is when they are going to culture in a culture medium. So what will be happen there? First, this recombinant DNA is going to what? Multiply. Okay. Then when they are going to introduce into this host cell, so in the host cell also in the new cell, by using the new cell, it going to what? The replicate and multiply antibiotic resistant genes in the way E. coli bacteria. So this is a brief introduction about the construction of first artificial recombinant DNA. Is it clear? Very simple it is. Stanley Cohen and Herbert Boyer. So these two are scientists belongs to United Kingdom. So these two scientists first constructed the artificial recombinant DNA. They first isolated the antibiotic resistant gene from the bacteria Salmonella typhimurium and then they are going to insert into the way vector DNA. That is vector DNA and that is called as recombinant DNA or chimeric DNA or mosaic DNA or hybrid DNA. Is it clear? And then that they are going to introduce into which bacteria? E. coli bacteria, Escherichia coli. So this one is going to what? Replicate to multiply the antibiotic resistant gene in E. coli. Means cloning of, then what will happen? It produces the multiple copies. That is called as the cloning of antibiotic resistant gene in E. coli. Cloning of antibiotic resistant gene in E. coli. Is it clear? So this is a brief introduction about the first artificial recombinant DNA technology, and after this we'll move to the next one. 3 basic steps of moving to the next one is here. Three basic steps of So there are how many basic steps of genetically modifying an organism? There are 3 basic steps of genetically modifying an organism. First one is here. What is the first step of the genetically modifying an organism. First one, identification. First step is identification of DNA with a desirable gene. Is it clear? Identification of DNA with a desirable gene. First, we have to identify the DNA with desirable gene. Second one, introduction. Second step is here, introduction of identified DNA into the where? Host. Is it clear? First, we have to identify the DNA. See here, we have to first identify the DNA with desirable gene. Okay, then, so this DNA we introduce into the where? Into the host cell. Introduce into the host cell. Is it clear? Moving to the third step is maintenance of introduced DNA into the host, then transfer into progeny. Moving to the third step is maintenance of introduced DNA into host, then transfer to where? Progeny. Is it clear? Maintenance of first one, identification of DNA. We are going to select the DNA. Which DNA? Desirable gene. Okay. Second one, that one we are going to introduce into, already I showed you diagram. This is the DNA. Okay, introduced into the where. So this one we are going to introduce into the host organism. After that, what we have to do? Maintenance of introduced DNA into host and then transfer to its progeny. Maintenance means what? Means we have to provide particular laboratory condition. Is it clear? We have to provide a particular laboratory condition. Is it clear? So that condition we have to what? The provide to this. So that is called as the what? Maintenance of introduced DNA into the host and then transfer to its progeny. Is it clear? So these are the three basic steps of genetically modifying an organism. Remember it. Even this is a very important question for the theory also for 3 marks. Is it clear? Mention the steps of genetically modifying an organism or mention 3 basic steps for genetically modifying an organism. Is it clear? Or even in the NEET they are going to ask the sequence. So you have to remember this correct sequence. Is it clear? So after this we'll go to the next one is to the tools of recombinant DNA technology. So what are the tools? Tools of recombinant DNA technology. First one is here, enzymes. First one is the enzymes. So what are the enzymes required for the recombinant DNA technology? That is the restriction enzyme. Second one is the ligase. Then next enzyme is here the polymerase enzyme. So what are the enzymes which are required? Restriction enzyme, ligase, polymerase enzyme. Let me try to explain about that one by one in detail. Second one is here, require the host cell. It also require the vector. It require the foreign DNA, require the bioreactor. So what are the tools of recombinant DNA technology? The tools of recombinant DNA technology are the restriction enzyme. Just now I told, again after this, we are going to study in detail about this restriction enzyme. It is already, I told that this is called as the molecular scissor because it cut the DNA at a specific region or the specific point. Second one, ligase. It is required for joining the two cuts end of the DNA. Second one is here the polymerase enzyme. So polymerase enzyme, it is helpful for the polymerization of the DNA or the RNA. Polymerization of the DNA or the RNA. You know very well, in molecular basis of inheritance, you are studied about the replication of the DNA. Then even I think maybe you are studied about the reverse transcriptase. Reverse transcriptase as well as the DNA polymerase enzyme. So this reverse transcriptase enzyme, reverse transcription means what? What is meant by reverse transcription? What is meant by the reverse transcription? Reverse transcription means here the synthesis of DNA from the RNA. Is it clear? The synthesis of DNA from the RNA is called reverse transcription. That is catalyzed by the enzyme is reverse transcriptase. You know very well, always RNA synthesized from the DNA. Central dogma of molecular biology was studied that RNA synthesized from the DNA. The biosynthesis of RNA from the DNA is called as transcription. That is called as the transcription. The synthesis of RNA from the DNA is called as the transcription. Is it clear? But in some viruses, what will happen there? synthesis of DNA from the RNA, that is called as the reverse transcription. That is called as the reverse transcription. Is it clear? So, with the help of this enzyme, reverse transcriptase, for example, you have to take down HIV virus. It is there. What kind of the transcription? Reverse transcription. Is it clear? Reverse transcription was discovered by the scientists. Who has discovered, children? It was discovered by the scientists Kevin and Baltimore. Kevin and Baltimore. Kevin and Baltimore. Kevin and Baltimore. Is it clear? So, here, second one is here the even in replication you have studied that two DNA strand. DNA is a double strand. Is it clear? One is 5 prime to 3 prime, another one is the 3 prime to the 5 prime. When hydrogen bond break down, what will happen? Two DNA strands are going to what? Separate. Is it clear? Two DNA strands are going to what? Separate. Now, both the DNA strands are acting as what? Template strand. Both DNA strands are going to act as what? Template strand. Was studied in replication. Understanding here? So both the DNA strand acting as a template. This is old strand. This is also old strand. So this template strand is going to produce what? One new strand, and this second template strand is going to produce what? New strand. One old, one new strand is formed. This is the new strand. This is the new strand. This is the blue color is old strand. So synthesis of DNA from the DNA template with the help of the enzyme. Which enzyme is required? DNA dependent DNA polymerase. DNA dependent DNA polymerase, and I very important I forgot to tell when I was telling you about antibiotic resistant gene, it will be replicate with the help of what? DNA polymerase enzyme. There I forgot to mention that one is here the this antibiotic resistant gene. So it is going to multiply with the help of the what? The DNA polymerase enzyme. So synthesis of DNA. Synthesis of what? DNA or DNA strand is catalyzed by the enzyme DNA polymerase or DNA dependent DNA polymerase. Is it clear? So these are the what? The brief introduction about the enzymes. Second one is here the host cell. Host cell. So host cell is also required for the recombinated DNA technology. So that is the what? The E. coli bacteria is one of the most commonly used bacteria as a host cell. Second one is the vector, that is the called as vehicle plasmid. Okay, and even for introducing a host cell, what we require? We require the foreign gene. What we are going to insert into the host cell, we are going to introduce a desirable gene, that is the what? The foreign DNA we are going to insert. So this is also required. Then last one is here the bioreactor. Bioreactor, it is a stainless steel vessel. It is a stainless steel vessel. What is the bioreactor? Bioreactor is a stainless steel vessel where biological reactions are taking place. Is it clear? Bioreactor, it is one of the stainless steel vessel designed to carry out biological reaction. In detail, you are going to study in this chapter only. Last heading is bioreactor only. Types of bioreactor, what is a bioreactor, what is the volume of bioreactor, that you are going to study in this chapter only. But bioreactor is a stainless steel vessel designed to carry out the biological reactions. Is it clear? Okay. So these are the tools of recombinant DNA technology. So what are the requirement? Enzymes such as restriction enzymes, ligase, polymerase enzyme. Even in some of the reference book, they are given that one is the alkaline phosphatase. They are given the alkaline phosphatase, so that in some reference book they are given as per the NCERT. They are not given alkaline phosphatase. So this alkaline phosphatase, it removes the phosphate. It removes what? The phosphate from the DNA molecule. So alkaline phosphate, but as per the NCERT they are given restriction enzyme, ligase, polymerase enzyme, E. coli host cell, bioreactor, vector. Is it clear? So these are the tools of recombinant DNA technology. Okay. Now after this we are going to study in detail about these enzymes. Okay. Moving to the first one, that is the restriction enzyme. In 1963, scientist Arbor. In 1963, scientist Arbor discovered. In 1963, scientist Arbor discovered that E. coli bacteria is able to protect itself from the bacteriophage. Understand here? In 1963, scientist Arbor discovered that E. coli bacteria is able to protect from bacteriophage. Protect from bacteriophage with the help of defense mechanism. Defense mechanism. It is called restriction modification system. Restriction modification system. Understanding here? In 1963, scientist Arbor discovered that E. coli bacteria, Escherichia coli, this bacteria, it protects itself from the bacteriophage. You know very well what is the bacteriophage. What is the bacteriophage? Bacteriophage is what? The virus which infects bacteria. Virus infects bacteria is called as bacteriophage. Is it clear? Virus which infects bacteria is called as the bacteriophage. In previous class, you are studying about the bacteriophages in lower classes as T2 bacteriophage, as well as the T4 bacteriophage. T4 bacteriophage you are studying. T indicate what? Tadpole-like structure. So there are the different types of the bacteriophages, but most common T2, T4 bacteriophages. In first year you are studying about this T2 bacteriophage, structure of the bacteriophage you are studying. Head, neck containing collar, sheath, tail fibers, pins. Is it clear? That is the what? The structure of the bacteriophage. So there are two types of life cycles of bacteriophage. One is the lytic and the second one is the lysogenic. That is actually not necessary here. Just to concentrate that in the year 1963, Arbor discovered that E. coli bacteria is able to protect itself from bacteriophage with the help of the defense mechanism. It will be defend itself. Defense mechanism is called as the restriction modification system. Then how this E. coli bacteria is going to protect itself from the bacteriophage? There are the two components are there. How this E. coli bacteria it is going to protect because this E. coli bacteria it contain two components are there. Two components are there. What are those two components? What are those two components? One is restriction enzyme. Okay. Another one is methylase. Another one is the methylase. Restriction enzyme is called as the restriction endonuclease also. Restriction endonuclease also. Restriction endonuclease also. Is it clear? So the bacteria has two components. These two are present within where? Within the bacteria only. Understand here? Scientist Arbor, he discovered that the bacteria protect itself from the bacteriophage with the help of what? Defense mechanism. How it is going to protect itself from the bacteriophage? Because it contains the two components are there. What are those two components? So those two components are the restriction enzyme and the second one is the methyl group methylase. Is it clear? Restriction enzyme it is a nuclease. It is a what? It is a nuclease has particular palindromic sequence. It has a particular palindromic sequence. It has a particular what? Palindromic sequence. Just before actually here just you have to take. I'll give an example. GAA TTC 5' to 3'. This is the 3' to 5'. So the sequence of the DNA, which is the same when you read in both the forward as well as the backward direction. How we are going to read forward direction? GAATTC. Backward dash, how we are going to read? GAATTC. The sequence of DNA is the same when you read in both the forward and backward direction. That is called as the palindromic sequence. Is it clear? After this introduction, I am going to give you definition of the palindromic sequence with example. So this is one of the sequence of the EcoR1, Escherichia coli strain 1. Is it clear? It is one of the restriction endonuclease. The name of the restriction endonuclease is what? EcoR1. So its sequence is this. Is it clear? So restriction enzyme is a nuclease has a particular palindromic sequence. Is it clear? So this restriction, why it is called as the restriction enzyme? Because this enzyme restrict the propagation of the bacteriophage. It restricts what? The propagation of what? Bacteriophage. So it is called as the restriction enzyme. Understood here? Restriction enzyme, it is a nucleus and it has a particular palindromic sequence. It is called restriction enzyme because it restricts the propagation of the bacteriophage. Propagation of what? It restricts the propagation of bacteriophage in the bacteria. Is it clear? So it is called as the restriction enzyme. Second one is here the methylase. So this one it is going to add what? It's going to add what? Methyl. It's going to add what? Methyl group. CH3 group. It is going to add what? The particular region. It's going to add the methyl group in a particular region. It has also a particular palindromic sequence. And this methyl group, when it is added here, so what will happen? So this methyl group, it protects the bacteria from this restriction enzyme. Is it clear? This methyl group is when it is added here, what will happen there? What will happen? So this is going to protect the bacteria. It protects the what? Bacteria from this restriction enzyme. Is it clear? So only they called that the bacteria is scientist Arbor. He discovered that bacteria is going to protect itself from the bacteriophage with the help of defense mechanism because of presence of what? Two components. One is the restriction enzyme, presence of restriction enzyme, and the second one is the methylase. Is it clear? Okay. Moving to the next one is the coming back in detail about this restriction enzyme. So restriction enzyme, it is called as the restriction endonuclease. It is called as the restriction endonuclease. Okay. So this restriction endonuclease, it is called as the molecular scissor, or it is called as the cleavage enzyme. Cleavage enzyme. So the first restriction endonuclease is Hind2, which is the first restriction endonuclease. The first restriction endonuclease is Hind2. That is Hind2 means what? Haemophilus influenza. Haemophilus influenza. The first restriction endonuclease was discovered by the scientist Hamilton Smith. Scientist Hamilton Smith, he first discovered the first restriction endonuclease, that is the hint2, that is the what? Haemophilus influenza. Is it clear? Hint2, it cut the DNA. It cut the DNA at a particular point. Hint2, it cut DNA at particular point by recognizing sequence of 6 base pair. Recognizing sequence of 6 base pair. Is it clear? So it means that the first restriction endonuclease is a hint2, and this restriction enzyme, it cut the DNA at particular point by recognizing sequence of how many base pair? 6. Remember, it is a very, very important point for the NEET. So recognizing sequence of 6 base pair, not 5, not 3, not 1. Is it clear? So after that, many more restriction endonucleases were discovered. Today there are 900 restriction endonuclease which are obtained from 230 strains of bacteria. 230 strains of bacteria. Is it clear? So how many restriction endonucleases were discovered? There are 900 restriction endonucleases which were obtained from 230 strains of bacteria, and each restriction endonuclease have the particular recognizing sequence. Is it clear? Each enzyme has a particular recognizing sequence. It means that each enzyme is cut at a specific region. Is it clear? So how many restriction enzymes are there? 900. So these are obtained from 230 different strains of the bacteria. Okay. Moving to the next one is here the what is the nomenclature of restriction endonuclease? Nomenclature of restriction endonuclease. The first letter represent the genus. Okay. Next two letters represent what? Species. Represent the species. For example, you have to take one more example beside the hint2. To take example is EcoR1. EcoR1. Here, EcoR1 echo R1. Here E is what? E is a genus. E represent what? Escherichia. It is a what? Escherichia is a genus. Okay. Two more letters. What are those? CO. It represent what? Coli. It is a what? It is a species. It is a what? Species. Is it clear? So here, the first one is here, the Escherichia. It is a what? Genus. And next two letters represent the what? Next how many letters are there? Two letters which represent what? The species. And fourth one is here, the R is there. If strain is there, then you have to write. That is the fourth one. This fourth letter represent the what? Fourth letter represent the strain if present. Rough strain, smooth strain. If it is a present, then the fourth letter will be the what? The strain. Then the next, it is followed by the what? Roman letter. Followed by Roman letter. Roman letter. If it was first, then we have to give the Roman letter 1. If it is going to discover second, then they are given 2, Roman letter 2. If it is discovered the third, then we are going to give the 3. So restriction enzymes maybe 1, 2 or 3, not 4, 5, 6, not there. Is it there? Only 1, 2 and 3. So here the first letter represents the genus name. For example, echo R1, E represents Escherichia, it is a genus. Second or subsequent two letters which represent what? Species. And fourth letter strain is there. That is the fourth, that is the R here. It is the R represent what? The strain, and followed by the what? The Roman letter 1, 2, or 3. Is it clear? So full echo R1 means Escherichia. Escherichia coli RY13. Actually, it is a full. You have to remember this is the echo R1 means Escherichia coli RY13. So this is the nomenclature of the restriction enzyme, that is the echo R1, and echo R1 or Escherichia coli RY13 also you can written. Its sequence is GAA TTC TTC 5' to 3', 3' to 5'. So this is the sequence of what? The echo R1. Remember, this is very, very important. The sequence of what? Echo R1. So in next class I will give the sequence of the hint 2 Haemophilus influenza. So hint 2, that sequence, and today I did not give. So in next class I will give, and I will explain you about the nucleases. What are the two different types of the nucleases? Then I am going to explain you about the palindromic sequence. Then the steps of recombinant DNA technology by the action of enzyme, restriction, endonuclease. Okay, thank you.